And we'll move on to our next talk. This is from Juliet Chu, and the title is Genetic Recombination in a New Magic Population of Peanut. So Waleed, stop sharing your screen, and then Juliet can share hers.
Hello.
Hello, Juliet.
Hi. Okay, let me start to share my screen.
Okay.
Is it shared?
Yep. So if you want
Okay.
to put it in presenter mode.
Yes. All right. Can you see it?
Yeah, perfect. There you go.
Okay, thank you. Okay, so this research is in collaboration with Dr. Josh Clevenger, Waleed, as he just presented, and Drs. David Berdioli and Peggy Ozias-Aikens from our research group from University of Georgia, and Dr. Cordy Holbrook from USD ARS in Tifton. So the MAGIC population stands for multi-parent advanced generation intercross population. So as the name indicated, this population consists of multiple parents. And then with multiple parents, the unique divergent parents would bring in more genetic diversity to the population. And also in the process of constructing the population, and several rounds of crossing will be done with a carefully designed funnels so that recombination would occur actively through several generations of crossings, and increasing the shuffling of the genetic diversity would be shuffled intensively in the process of population construction. So in the end, the MAGIC population lines would host fine-scale mosaic allele contribution from all founder parents, but that's the theory behind using multi-parents to produce a genetic mapping population. And because of this process, this mapping population is really desirable to perform high-resolution genetic mapping or GWAS analysis. This is the benefit of having the MAGIC population, but the downside of producing this MAGIC and to utilize it is the large effort needed to do the construction, to advance the population, and also to genotype and phenotype the population. So with the funding from NIFA, our group were able to initiate the construction of a new peanut MAGIC population consisting of 18 founders. And here is an image of the seeds from all founders. And you can already see that they are highly diverse in the selection in regards to their seed characteristics, like their seed size, seed coat, and also because this is consisting of both cultivars and germplasms. So there's a diverse range of yield also hosted in this founder range. And then the selection of this MAGIC population also take into consideration of those important diseases that threatens peanut production. So here we have the founders have unique contributions to resistance to TSWV, early spot, lady spot here, and cylindrocladium black rot, Scrotinia blight, white mold, rust, smut, and also drought tolerance and aflatoxin resistance to aflatoxin contamination. And these founders also covers all 4 market type of peanut. So with this highly diverse panel of parent inclusion, this population upon construction can be used widely to fine mapping of all these important traits that would increase peanut yield and protect its yield potential by fine mapping those disease resistance. So here describe the process of this MAGIC population construction. So as I said in the earlier slide, you have seen the seed images of these founders. So between this, among these 18 divergent parents, we made random crosses among the pairwise crosses as the initiation of the construction. So the two-way crosses, there were 9 two-way crosses that has been performed by random pairing these 18 divergent parents. And by 2019, we had recovered 129 F1 hybrids. So this whole process of magic population production, we genotype all the founders and also all the potential hybrids recovered from each step of process. And so the hybridity of the hybrids were confirmed through genotyping. And then upon the recovery of the F1 hybrids from the two-way crosses, we proceed with 36 four-way crosses following a half diallele mating design. And by the end of last year, we recovered 186 four-way F1 hybrids. And then we proceed with 72 eight-way crosses that were designed to balance the allele contribution from all founders. And now, just last month, we recovered 266 eight-way F1 hybrids. And all these hybrids will be genotyped either through the array or using Caspar assay. And then for those true hybrids, we're going to proceed with 16-way crosses to stack most of the founder alleles in every line. And we are targeting is ambitious, but we'll try to do it. We're targeting to recover 700 to 1,000 16-way F1 hybrids by next year. And these F1 hybrids is going to be go through single seed descent to by speed breeding in the greenhouse. And then hopefully our population can be established by year 2024. So here shows you the specific hybrids recovery from the two-way crosses and then the combination of this random pairing of the 18 founder parents. And this is a four-way design. It's a half diallele mating design. And then each combination and the F1 and the F1 recovery. And this is the eight-way cross design because this all the highlighted cell indicating possible combination to for all the two-way F1 parents. But because of the limitation of F1 hybrids recovered from all the combinations, only the green highlighted crosses were made it to the four-way cross. As I said, all of our material were genotyped using the version 2 Arachis SNP array. So for the next slides of the data, we use a polyhybrid resolution class of this array because it's presenting the best quality data. In this material that we also have, because of disease reasons, we also have known sources of alien introgression harbored in those founders. And those are from historical, the breeding effort through many groups, particularly the Texas and also the North Carolina groups that they introduced the cardenasii resistance into that actually Dr. David Bartioli talking about part of this as well. The IC322 from Brazil hosted introgression from the A02 and A03 region from cardenasii, and Bailey has A08, and Tiffany Hallel has A09 that actually was from Texas effort. And Dr. Bartioli did analysis using his diagnostic markers for those introgression and to look at our four-way F1 hybrids. So this slide shows you the introgression. So horizontally, each column is an individual F1 hybrid come from the four-way cross. And each row of this very miniaturized table is the SNP markers that are diagnostic for the introgression that has been ordered by the chromosome positions. So here the highlighted are the introgression specific markers that for each individual. So you can see in these all of these four-way F1 hybrids that actually that have different combinations of those introgressions that are from A02, A03, A08. All of these regions have been shown to have a contribution to leafspots and rust resistance. And for A09, there is a known introgression that confer nematode resistance. So all of this has been integrated or mixed in our F1 hybrids. So the potential utility is a spin-off from MAGIC, is that from this recombinance with allele introgression, is that these lines with various combinations of introgressed segments can be used for further defining the disease resistance associated with these segments. Because this is only F1 of the four-way, we actually advance these materials, and then maybe further recombination could happen, and this could serve as a purpose for defining the specific region for the resistance. And then lines permeated with multiple segments can be evaluated for cultivar release, or they can serve as valuable genetic sources for breeding programs. And the next thing we study is because as I said, the whole concept of constructing the MAGIC is to introduce high rate of recombination among the lines to provide the genetic basis for high resolution genetic mapping. So we are interested in recombination rate. So we take a pilot study to look at the recombination events in the four-way F1 hybrids. So the way we did it was to filter the genotype data by retaining the markers containing the unique alleles from one of the four founder parents. So here's an example of how this has been done in a specific, like one specific marker. So in this case, the example is a CC477 has a unique allele that is different from the rest. The three parents are used for this four-way crossing. So in the beginning, like in the initial two-way cross, and then because we assume this homogeneous in this locus, and you will have the hybrids after the two-way cross to have this CG, which will be called as AB, is here. I don't know if I can see it. Yes, you can, right? Yes. So it would be called as AB because you have CC477 bringing the unique allele. That's a different and then here shows you this particular individual is a two-way F1 hybrid, is calling as AB. And then another, the counterpart or serve as male in the next round of four-way cross would have a GG because it's a common marker. So here you would say like a BB call in this actual case. But then the meiosis happens during when we use the two-way F1 serve as male and the female for the four-way cross. In this case, when the meiosis occurred, forming the gametes for the egg cell and pollen. So in this case, after fertilization, this recombination would occur. I mean, the shuffling would actually happen in this stage. So you would recover like the random combination. You would have equal number of individual with a CG or GG combination, or AB call and BB call. So here you can see actually we have 6 four-way F1 being genotyped here, and yeah, they do have like equal number of BB and AB call. So when we think about this in the border scale, like look at the recombination when we consider a chromosome. So in the beginning, the two-way hybridization allows the merging of the divergent parents into one individual, one F1 individual. And then actually our first round of genome shuffling occurred during the formation of the gametes for the four-way crossing. So this meiosis in this process of the formation of the gametes actually introduced the recombination. And then when the four-way F1 formed after fertilization, the recombined segments merged into the four-way F1 cell. So with this concept, we determined to count the recombinant blocks in the two parents of four-way cross separately, because there are two sets of recombination occurred when the egg cell and the pollen cell were formed. And then the counting method was developed by Dr. Walid Korani using the R scripts. And then the number of recombination events from the two sets of counting were added together to account for the actual number of recombinant events in the four-way F1 hybrids.
Under 2 minutes, Juliet.
Huh?
Under 2 minutes.
Okay, thank you. So here shows you the heat map of the recombinant blocks from female-specific unique alleles, and then the male unique, and then the common blocks. When we add them together, this is a total recombinant events that can be counted in this way for the four-way F1 cross. So in summary, we are under the construction of a new MAGIC population consisting of 18 founder parents with highly diverse genetic traits and active recombination events of F1 hybrids from two-way and four-way crosses were captured, and tracking the combination segregation of the known introgression segments facilitate the foreground selection of lines with multiple disease resistance. And recombination events captured in the four-way hybrids exhibit the great potential of high-resolution genome mapping with progenies from this project. So I would like to acknowledge the members, my colleagues in Dr. Peggy Aiken's lab, Stephanie, Kathleen, Micah, Chandler, and Kamal Prait, and our collaborators, Dr. David Bertioli, Cordy's group, Shannon and Jason, and of course, the contribution from Dr. Josh and Waleed. Our founding agent is NIFA. Thank you.